chemistry, medicinal
Okay.
chemistry, right? Ma'am Suchita, ma'am, can you unmute yourself?
Yes, sir.
Unmute, ma'am. You please
Oh,
unmute
sorry.
yourself. So over 23 years of experience. So I was guided. I was supervised 10 of the PhD students and more than 70 pharmacy graduates and around 120 B pharmacy graduates about their project thesis, right? And presently, six of the PhD students under working under my guidance, right? So basically, already I said my background is pharmaceutical chemistry, and I do my uh, intra, and multidisciplinary research, right? So that is why my research area of expertise is nanotechnology. It means nanotechnology and delivery, nano herbal formulations, then drug design and discovery through these in silico studies, then method development and validation. So that is comes under pharmaceutical analysis, right? Using HPLC and LCMS and UV, etc. And then I also know, so carry out with the support of my team members about this pharmacology biological evaluations of the synthesized nano herbal formulations. And then I also carry out some research in the medical devices in the area of medical devices. So recently we have developed one biosensor device for the diagnosis of breast cancer. Uh, it is already filed a patent, right? And I also do some designing the apps because we are the basically background of pharmacy. So healthcare, we are also part of healthcare professionals, so that we have the background area of the diagnosis and ADRs reporting and drug interactions and those related issues, economics, genomics, these are all those things, so that we also can take the support of PharmD guys to in the design and development of medical apps, right? So two medical apps also I was designed, had heart care risk assessment app one and to assess the thyroid activity. So these things, it means intradisciplinary and multidisciplinary. So when you go for the medical devices, design and development and medical devices, you should have a support of engineering guys, right? And whereas if you likewise the app designing also engineering guys support is required. So that's why I'm talking about multidisciplinary research. Yes. So I think this is a brief thing I wanted to enlighten to the faculty members, because faculty, we are all faculty members. We know the importance of research. So every teacher, being a teacher, we deliver the lectures to the students and we take the classes, theory classes, and we take the lab reports. This is the primary role of any teacher. And in addition to that, at present, we have the targets for accreditations and rankings. For this, the faculty member also be no, so they have the strong contributions and background in research, right? It means based upon our research contribution. Suppose you take NIRF ranking, 30% of weightage is given to the research. It means publications, grants of the patents and publishing patents, then citations of the publications. So 30% weightage will be there. Like that, around 40 to 50% weightage is there in the global rankings. It may be QS ranking and THE rankings. So that my request here to all the faculty members are, so you be focused on research also in addition to your regular. So everybody is having workload about 14 or 15 hours, but whoever is working beyond, like additional right? And taking additional like portfolios, or you are spending time in the research. So definitely you are leading going to get a incentives, else you are going to have the some positions. Okay, so like that. So this is my suggestion to faculty members, right? So be strong in your research also. Okay. So with this brief introduction to the faculty members, before going to this in-depth discussion on today's title, I request everyone please take a small observation notebook, any rough notebook, and note down the key points. Okay, whichever looking important for you, you can take the inputs from this, right? So every session you will get some outputs, right? So those outputs definitely useful one or other day in your future, right? So now I am moving to my today's session. Today is day 4, and the session is online international FDP. We are carrying out with the collaboration of University of Cyberjaya, Malaysia, and the no, Equipline Technologies Private Limited. It's a pharmaceutical company. So we collaborated with them, and we have started working on this, and our journey, no, 3 days journey is completed. This is 4th day. And today I am here as a resource person to deliver this session on design and development of nano formulations, nano herbal formulations, and its evaluation process, right? So this is the title, how to design and develop nano herbal formulations. Here, if you can go through the slide 1, that is the title slide. Here, I was mentioned the SDG goals, Sustainable Development Goals. We have 17 Sustainable Development Goals, right? So whatever research we are carrying out, right? Whatever developments we are doing in our country, that has to be beneficial to the society and public. End of the day, we will be recognized when our research, right? So when our contributions are beneficial to the society, so then only right, our dreams are going to be fulfilled. So that in this context, whatever research you are carrying out, you should target to the sustainable development goals also, whether it is beneficial to the public or not, society or not, or our country or not. So that now that is why QS global rankings, they have this year onwards, they are about to release along with the QS ranking, world ranking, they are also releasing sustainable development rankings, okay, to the universities. Like that, THE already past 2 years, they are releasing ranks for sustainable development goals. That's why whatever brochure we are preparing in the campus colleges, we should keep any goal which is matching to our presentation, or it may be a event. Okay, it may be your review or research articles. So you should know keywords should be mentioned. In brochure, we will mention the logos. Okay, so here with this session, today's session, good health and well-being, because I am talking about the healthcare related things, right? So that that is why it is fulfilling third goal of the sustainable development. And the quality of education, you are taking you are taking some key points, right? So yeah, from this session, so that definitely you are getting some inputs. It's why, that is why it's a quality of things, right? And we are collaborating with the industry so that it is fulfilling ninth goal, and partnering with the Cyberjaya and other university, other pharmaceutical industry, so that these four goals are matching with this my presentation. So that I was mentioned this, right? Now move on to the nano herbal formulations, right? Here, what is meant by nano herbal formulation? Nano herbal formulations, it means they enhance the therapeutic property of herbal medicines by utilizing nano technology, right? Okay, so here, what is nanotechnology, and what are the different types of nano formulations are available? All those things will come in next slides. But here, nano herbal formulations, mainly, generally herbal formulations will be there. But here nano herbal formulations and herbal formulations, what is the difference, right? So those things I am going to highlight in this presentation. In this context, main key objectives of this preparing nano herbal formulations is to enhance the solubility and bioavailability, because these natural products, so solubility, because herbal formulations mean it is a bunch of constituents are present in the each extracts or powder. Okay, so this bunch of constituent to dissolve, so it is no toughest task to the research scientist. So in this context, these nano formulations are helping us to enhance the solubility and bioavailability of natural products, and stability is also be enhanced with this nano herbal formulation, target delivery, and we can reduce the toxicities and side effects, and we can enhance the therapeutic activity also. Okay, so these are the advantages we have with this nano herbal formulation. So previously we come to know about the herbal formulation. Okay, these are the drawbacks of herbal formulation. To counter the drawbacks of herbal formulations, we are going for the preparation of nano herbal formulations or phytozoans. Okay, now you see why to go for herbal formulations. So why to prepare and develop these herbal formulations? Because the natural products, still daily whatever food materials we are taking, they are all natural products. You know that. Okay, natural from the animal resources or plant resources or no sea marine resources, right? Compared to synthetic, we are taking natural products only daily for our livelihood. We take this one. So those food materials, knowingly, unknowingly, beneficial to all of them, like to clear the mental or nervous disorders, cardiovascular diseases, antimicrobial properties, inflammatory related, cancer related. So all those things, whatever food materials we are taking, either vegetables or fruits or okay, so leaves, whatever maybe it is, whatever things we are taking, okay, or carbohydrates or proteins, whichever we are taking. So those are definitely what is happening is going to be beneficial. Okay, so that's why we have to prepare the nano herbal formulations, right? Yeah. So now, Sajji, वो डोर बंद कर दे। वीडियो और कर दो। कैमरा सब कैमरा ऑन करिए। Yeah. So, Mamta ma'am, am I audible, ma'am?
Yes, sir. You are quite audible.
Okay. Sorry for the interruption. Now, we are moving on to the next slide, right? So this is and all. Previously, I told you why to go for the nano-herbal formulation, because we are having so many benefits from these natural resources. So that we need to have these products, right? And coming to the just here, I am highlighting that because somebody may think that why to go for the nano, why not synthetic? Okay, synthetic also we can go, but these natural products also we are having better therapeutic activities. So that here, here for carrying out the research work, if you have a no wide range of activities and actions, then we can enter into particular research. That's why I'm here highlighting the therapeutic applications of herbals actually. So there is some herbals which are in the treatment of cancer therapy. So for from like Thymine vulgaris and Senna Alexandria. Then we some of the herbals are using in the treatment of inflammatory and neurodegenerative disorders. For example, ginger, curcuma, boswellia, green tea, licorice. These are all no using in the inflammatory and neurodegenerative disorders. So some herbal formulations in the cardiovascular health, garlic, Google, then green tea, ginkgo biloba, terminalia, onions. These are all using in the cardiovascular health. Skin problems, right? And again, aloe vera, neem, curcumin, chamoli. These things are using in this treatment of skin-related problems. So some of the like natural plants also beneficial for these antimicrobial antioxidant applications. Oregano, oregano, rosemary, thyme, cloves, cinnamon, garlic, medicinal plants, then turmeric, like this. And we have other applications also with this herbal formulation in the agriculture, food industry, cosmetics, right? And some marketed nano herbal formulations also available now. And then artemisinin, everybody knows that. Artemisinin is no anti-malarial agent. Okay, some curcumin nano carriers also we have developed. So these are the some just you see these are the formulations we are using and the benefits also telling, so that we have to carry out extensive. There is a possibility of carrying out extensive research in this nano, not nano. So these herbal natural plants related area. Now coming to the my next part of my presentation. Now coming to the what are nano preparations. Everybody knows that nano size or nanoparticle is a one of the special class materials that enclose particularly substance which has not done. So nano means the particle size of the drug molecule is no very small, right? Like 1 nanometer to 100 nanometer size. So when we are preparing this nanoparticle size, what is the benefits? Everybody knows that if our particle size is reduced, the surface area is increased, solubility is increased. So obviously bioavailability of the drug also increases. So that is why we are preparing nanoparticulate materials. Understood my point, right? And coming to the it is nano research is no presently the wide range of revolution and developments is happening in the field of nanotechnology, the treatment side, monitoring, diagnosis. Okay, so in nanotechnology, also in the computer field area, right? Microchips designs, then drug delivery systems. Okay, so everywhere these nanotechnology now applying, right? In our case, the particle size is decreasing because of the to enhance the solubility and to enhance the bioavailability of particular materials. Yeah. So here, what type of herbal drugs? Why we should go for nano preparations for herbal drugs? Herbal drugs means plant resources because of having low absorption. So absorption is very low. That is why we have to design and develop this nano particulate material and inability to cross the biological membranes. So when we take the nano formulation to cross the blood brain barrier or to cross the lipid biological membranes, So it is very much difficult for these direct complex or herbal drugs. So that nano formulations or phytosomes are preparing to cross the lipid membranes. So high molecular size will be there. So that is why we have to go for the particle size reduction. So poorly absorbed. So absorption is poor. That is why to enhance the absorption, these small particle sizes play vital role. Yeah. So now, what is happening? Already I told here. So materials, size, surface, and shapes. So based upon the nanoparticle size, its surface, and its shape, what is happening? Nano carriers are classified into two types of classes basically. So we are using to prepare these nanoparticle particulate materials. We are using organic carriers and inorganic carriers. Two types of carriers may be using, right? So we'll be using for preparing these nanoparticle dosage forms, right? So that is why here to enhance the kinetic property. Okay, so we are using some carriers and reacting, forming a complex with these carriers. These carriers are two types, organic carriers and inorganic carriers. Okay, you can select any organic carrier material depending upon your product, right? Or inorganic material. So based upon the literature review and structural similarity of the pharmacophore or your natural active constituent, we can select the organic nanocarrier or inorganic nanocarrier. Okay. So now coming to the different types of the nano formulations, we can call it as a liposomes, myceliforms, neosomes, solid lipid nanoparticles, nanogels, gold nanoparticles, carbon nanotubes, calcium phosphate nanoformulations, dendrimers, mesoporous compounds. Like that, we have different types of nano formulations. So this is the thing here. If you can see here, so these are the various formulations. Suppose in this slide, the nanoparticle size is 1 nanometer to 100 nanometer, right? The surface and shape and material, these things we need to consider. And how we are preparing these nano herbal formulations, either in a form of electrospinning technique, homogenization technique, nano emulsion form technique, nano encapsulation, nano fabrication, nano polymerization, any polymers we can, nano precipitation formula, nano suspension, spray drying. With this, any one of the technique, we can prepare these herbal nano formulations. Yeah. So coming to this slide, okay. So here, what is happening? So take a drug. You can select any nano carrier. Any nano carrier, it means any inorganic nano carrier or organic nano carrier you can select. Inorganic nano carriers means it may be a gold nanoparticle, calcium phosphate, so carbon nanotubes. These are all inorganic nano carriers. So these are all polymeric nano carriers means it is all organic nano carriers. These are through organic nano carriers. We will prepare myceliforms, solid lipid nanoparticles, nanogels, liposome. This is and all. So as in previous slide I said, that is the organic nanocarriers and inorganic nanocarriers. Through inorganic nanocarriers, we will develop some type of nanoformulations. Through organic nano carriers, we will develop some types of this one. Whichever no carrier is required to your formulation, you need to select that particular nanocarrier. Yeah, so these are all already we were discussed, and these are around 11 types of formulations, nano type of formulations we can design and develop. So among all the 11, any one nano formulation you can develop for your selected herbal constituent, either single active constituent or combination of active constituent or combination of herbals, combination of plant powders. Okay. So single we can prepare nano formula. Single means active constant, isolated active constant from the plant. We can prepare nano, or else so one plant contains bunch of the active constant. So we can prepare one particular plant powder nano formulation. Second one. Third one, so we can combine 2-3 plants, whichever showing better response, 3 plant combined, and we'll go for nano formulations. For example, polymeric nanoparticles, solid lipid, magnetic nanoparticles, metal inorganic nanoparticles, quantum dots, micellase, phospholipids, colloidal nanoliposomes, dendrimers. So like that, these are a variety of formulations we can design and develop. These are some images. How after preparing the nanoparticles, how these that formulation looks like, right? So it may be a 0D material or 1D material, two-dimensional material. Mesoporous means the solution is will be having like this liposomes, okay, and mycelium, polymeric nano. These are the no. So when we are preparing any type of formulation, it looks like this image. Okay, now how we will design, how we will develop these nano formulations. There is some technique. Suppose you have a active constant in your hand, you have a nano carrier in our hand, right? How we will form a complexation with this through some techniques to develop this particular nano formulation. Out of 10 or 11 nano formulations, you can choose any one of the nano formulation. We can go for some methodology for preparing the nano herbal formulation through hot homogenization technique, cold homogenization technique, complex co-observation technique, co-precipitation method, salting out method, supercritical fluid method, solvent emulsification diffusion method, nano precipitation method. So like that, we have 8 type of techniques. So 8 types of techniques we have, okay, methods of preparations we have to prepare the nano herbal formulations. Among all the 8, so depending upon your formulation, whichever formula you wanted to prepare, which type of nano formulation you would like to prepare, you need to select such methodology. So if you don't have any idea which method to be selected, you can go through the literature, vigorous literature survey, okay, and standard literature survey, okay, from reputed journals and articles. So such technique you can select for the preparation of this nano herbal formulation. Okay, so this is one of the image. So here initially we are having the plant. Okay, so this plant, then we are taking phospholipids. These phospholipids, and this is the lipid one, right? That is the carrier, and this is the plant resource, either the direct powder or active constant. Both of them forming a complexation, and finally your formulation comes like this. Okay, this is a herbal loaded nanoparticle formulation. Either single active constant formula or a bunch of active constants will be there like this, or else no bunch of plants may be combining each other, and we are getting the final end product like this. Okay. These are the methods I was highlighted, like hot homogenization technique, how we will perform hot homogenization technique, how we will perform cold homogenization technique. Okay. So cold means we have to melt the our powder with the lipid, and both of them, drug and lipid, both of them melt and quickly apply to the cooling conditions. Using liquid nitrogen and ice nitrogen, you will get a formulation. Okay, complex co-observation technique. These are the just mentioned the procedure, how we will prepare the complex co-observations, and precipitation method, how we will perform this precipitation method, what is meant by salting out methods, supercritical. Among all these 8 techniques, any one technique you can select for your preparation of nano herbal formulation. Okay, so this is the how this nano formulation is binding to the particular target enzyme or receptor or protein. How it is going to be bind? This is the process. So this is the like the nanoparticle. This is our nanoparticle immunoglobulins. That is when it is going to the blood. When it enters into the blood, nano formula. So it combines with the immunoglobulins present in the blood, and there is a complexation is formed in between nanoparticle and immunoglobulins. That process is a opsonization. Then these complex is binding to the particular receptor or protein present on the surface of the cell. Okay, so this is the phagocytosis process. How nanoparticle will act as a product, how it is binding to the particular receptor or enzyme or protein. Okay, so nanoparticle initially combining with the immunoglobulins and form a complex. This complex is binding to the particular receptor or enzyme. That process is called as a phagocytosis process. Now we have some equipments to develop these nano formulations. That is a sputter coater. This is one. This is the instrument image sputter coater and field emission scanning electron microscopy. Okay, these are the through these instrument we will we will go for the coating to the nanoparticles, or we'll prepare the nano formulations. Okay, photolithography, ellipsometer which is using to measure the optical constant and thickness of the nano formulation. Okay, these are the equipments to prepare the nano formulation and to measure the nanoparticle. It means nanoparticle surface area measurement, nanoparticle size measurements, nanoparticle shape measurement. So these things will be done through these equipments. Okay, so these are high-end equipments. Mostly most of the equipments we will find in IITs. Okay, so because they are even they are engineering guys or healthcare related people, they perform these nano formulations. Okay, so Raman spectroscopy and nano imprint lithography. Okay, and X-ray diffraction method. X-ray diffraction method, everybody knows that, right? So in X-ray, if there is a straight line, it is coming. It means your formula is a powder form. If peaks are coming, showing in the X-ray diffraction method, peaks, it means your particle is in the crystalline form. So there is a X-ray diffraction method is useful whether your formulation is in a powder form or in a crystalline form. If it is a crystalline form, okay, peaks will be coming from the X-ray analysis. Whereas if straight line you are getting, it means it's a powder form. Okay, so thickness we can measure, and fluorescence techniques. Yeah, these are the several equipments I was mentioned here, and these are the list of some of the equipments we will use, right, to prepare and to to identify, to confirm. So these are the equipments we will use. Okay, these are the n number of equipments we will use. Out of these n number of equipments, I will show you very few equipments, few results. Data is mandatory for our publication and for justifying our results. Okay, so where one is a like X-ray powder diffraction method. Okay, so it is a already I told X-ray diffraction method is useful to the whether it is a crystalline nature or your formulation is a crystalline nature or in a powder form, right? And another one is a thermogravimetric method. So out of all the previous slide, I was mentioned several equipments, right? So out of which, what are the most important equipments which are using to confirm the formulations and to justify our formulations, right? Here, first one is X-ray powder method. Next, TGA, thermogravimetric method. This is mainly useful. So here, what happens is temperature recording. For example, your active constant temperature is 120 degrees, right? So that active constant you have isolated from the plant. You have formulated nano formulation using some carrier, right? So there is any interaction was happened in between your selected polymer or nano carrier with drug. Suppose if complex is formed, you cannot get 120 degrees response in the DSC analysis or thermogravimetric analysis. Understand my point? So if interaction is not happened, you will get a response is 120 degrees. This 120 degrees you are getting a peak. It indicates that your active constant, okay, your active constant is alive. There is no interaction was happened in between both the your active constant and carriers what you have used for the formulation, right? So for that, either DSC or TGA analysis is important, right? Through this technique, we will determine the melting point of particular compound and glass transition temperature. It means you have through this nanotechnology, you have developed one glass material. Okay, you have developed one glass material. Whether this glass material is melted up to which temperature. Okay, so those things also we can measure because in thermogravimetric analysis or DSC, we can measure up to the 2000 or 1500 to 2000 melting point. Okay, so that much of melting point we will measure. For example, cement industries, okay, paint industries. Suppose you have uh, you have built the one okay, so home, right? So if any calamities are coming, high temperatures, 50 degrees, like that we are getting in the summer, right? So if 50 degrees, if it is dissolved, cement, whatever we are pasting, so then what will happen? So like that, it may be a cement industry or plastic industry or glass industry or our pharmaceutical industry. Okay, so everywhere the melting point of the particular sample or substance measurement is very, very important. Here in our formulation, interaction is happened or not, we have to check it out. Okay, or else in our formulation, our drug is alive or not, we have to check. If same melting point we are getting the response, it means our drug is not interacted with any polymer or any other excipients what we have used for the nano formulations. I think understood. Okay, so next one is to determine the particle size and the polydispersity index, right? This is also the particle size of the nano formulation is also important. For that, we have the equipment that is DLS. DLS is one of the instrument, dynamic light scattering method. This is and all in no reputed universities and IITs, such type of equipments are available. And no, we will send our sample to particular no place and we will get the results. This is also mandatory for publication and for thesis, PhD. DLS, that is dynamic light scattering method. That report is very, very important. It is useful to measure the particle size and surface charge and stability of particular nano products. And another one is a TEM analysis, transmission electron microscopy. So this is results also important. TEM results, DLS results are important, light scattering technique results, and TEM results also is important for your nano formulations or nano herbal formulations. So TEM is useful to determine the structural surface and shape of the carriers or your nano formulations. So dynamic already DLS already we spoke. The particle size is measured. And NMR. So NMR, nuclear magnetic resonance, is used for the estimation of the qualitative nature of the sample. So if it is there, drug and any complexation, no, nanoformulation means so many things are there. Whether to determine the physicochemical, it means structural changes, anything is happened or not. Okay, functional group, it is present or not. Okay, so for that, NMR results also is important. IR results also are important for the final conclusion and evaluation, right? Now, determination of encapsulation efficiency and recovery. So HPLC method and UV method also we can do, or LCMS method also we can perform for the quality or quantitative estimation of the product. So depending upon the response of the peak, we will determine how much quantity of particular X constant is there. How much, suppose in your nano herbal formulations, 5 constants are there from 3-4 plants. These 5 constants, we should know how much X constant is there, how much Y is there, how much, depending upon the peak response, right? In the HPLC or LCMS, we will calculate the peak area. Through that, the overall, so the percentage of particular constant is present. And in addition to the some more to check the stability studies, we need to depend on some techniques like UV method and TEM, zeta potential techniques like this. Okay. SEM data is also is required, okay, to understand the surface structure of the sample. TEM, SEM, DLS, okay, NMR, FTIR data. So for quantitative determination, quantity determination, HPLC, LCMS. Okay, these are the data are required for the confirmation of these nano herbal formulations. Now, so just I would like to give, because you are all faculty members or research scholars, I would like to give you one small input. How we will carry out these? So what type of nano formulations to be prepared? Because we have plenty of natural resources, plenty of marine resources, plenty of animal resources, right? So then how we will start the research work? Okay, suppose you want to design any herbal formulation. Okay, you want to develop any herbal formulation. How can you select a particular plant, right? So this plant I would like to prepare in a form of nano herbal. So how you can select the particular plant or particular constituent or bunch of plants? So based upon in silico studies, you can start your journey from the in silico studies, followed by the in vitro studies like animal studies. Then we can go for the phytosomes or nanoherbal preparation. So initially, how we can carry out these in silico design and development of particular active constant or plant products, right? So what you have to do is, right? What you have to do, for example, you want to carry out research work on any X plant. For example, you want to carry out on ginger. Whether ginger, it is really beneficial to reduce my lipid content in the body. Okay, whether it is reducing lipid content in the body or not. First of all, you have to assess. Okay, based upon your research work, then only we can go for the development of that ginger nano herbal formulation. Understand? So how we will start the work? Firstly, so take the ginger plant, and from the literature you can identify what are the active constituents really present in the ginger. Okay, gingerberry. Okay, so you have to identify the active constituent. That active constituent, right? You take the structure of that active constituent and go for the in silico studies, docking studies on. Suppose you want to carry out hyperlipidemic activity, you can take the PDB. Okay, protein data from PDB on IDs will be there. You can download the HMG-CoA enzyme. HMG-CoA enzyme is showing the anti-hyperlipidemic response. HMG-CoA reductase. For example, statins are there. You can compare with the standard like statins. Any like rosuvastatin, atorvastatin, simvastatin. You can take the standard and standard with the HMG-CoA. What is the docking score? And like that, your natural active constant, right? So ginger you have taken. So major active constants, major active constant only you have to take for the in silico study. Not all the constants. Suppose plant you can take 10 or 15 active constants will be there, but need not to take all the active constant. Really, what is the major active constant present in that particular plant? That major active constant, one or two major active constants, those active constants structures to be drawn first. Then we can go for the docking on the HMG-CoA, whether this ginger is showing our anti-hyperlipidemic activity or not by comparing the standard compound. So like that, you can take major active constant from the plant. Those active constant we can study. For example, Alzheimer's study you would like to carry out. Okay, so Alzheimer's study karna chahte hain, right? So I was mentioned NMDA receptor. So NMDA receptor you can download from the PDB And so suppose आप कोई मिर्ची पे काम करना चाहते हैं, capsaicin. Okay, so capsaicin, you take the structure and NMDA receptor docking करना. So standard drug, any donepezil, okay, or else any memantine, okay, so rivastigmine, any one standard you can select and you can start working on that. If you are getting a better results, better docking scores, best docking scores, okay, then what you will do is you can go for the next things, like docking scores are good, then animal studies followed by herbal formulations. So like this. Suppose after getting these docking scores and good results in animal studies, we can go for the patent publications also, commercialization also, like that. So our vision should be to reach to the society. So it means, so preliminary research, how to start our research work, and after that animal studies. After that, I am telling you nanoherbal formulations, how to design and develop what I discussed previously. Like that, these are the some enzymes or proteins I was mentioned over here for carrying out thyroid related activities. Nowadays, no, the women's, those who cross the age more than 30 years or 35 years, okay, around 25-30% of the women's, they are suffering with these thyroid problems. Okay, we should give, because thyroid, only 3-4 drugs are there in the market, right? So only 3-4 drugs only. I think everybody knows that. Okay, so we can take the active constant from the plant. We can go for the docking studies by comparing with the standard. Then we can give the better products to the society. Okay, so like that is there. For example, you want to carry out any active constant plant product, breast cancer activity, aromatase enzyme. Aromatase enzyme, we can download the aromatase enzyme from the PDV, right? And your structure active constant, both of them, right? So previously, whatever formulation it is available in the market, there is no that much of evidences. Now, particularly, this plant is showing some results from the literature review. Some X plant is showing the results. So you can start working like this, right? Go for the in silico studies and data. Better if results are good, then you can go for the next step, like animal studies followed by the final instance. And as I said in the right in beginning of the my presentation, like I was mentioned about the applications of all those things, right? Here, some therapeutic applications I was mentioned, like biological activities I was mentioned. But here, these nanoparticulate dosage forms, they are very much useful in the drug delivery related things and food preparations, gene delivery related, cancer treatment, cosmetics, industrial applications, right? Tissue engineering. Okay, so like that, in so many applications are there with these nano formulations. Okay, so these are the things I would like to share with all of you, right? This is my presentation. Then these are the some glimpse of the formulations, how it looks like. So these are all the, nowadays for cosmetics and all, most of them from natural resources only. From natural only we are preparing all these. You see these cosmetic products. So through these formulations, right? It is enhancing these skin performance and all like this. So these are the some glimpse I was mentioned here, right? So, and lastly, what I wanted to tell you is, everybody is looking for success. In beginning of my presentation, before starting my presentation, I told you that, okay, research contribution of the employee is definitely, it is a, it is a highly valuable to the universities or colleges, and to get the like incentives or positions, all those, depending upon your research contribution only. Understand? So that's why I request all the faculty members, those who are in this online session, right? You must start and must spend some additional time to recognize yourself by the employers. Okay, and to recognize here, second point. Second point is interdisciplinary, multidisciplinary. How it is going to be happen? So networking and collaboration. Okay, networking and collaboration. We can okay, so flourish ourselves and we can publish ourselves. We can go for the commercialization. So that is why I request the faculties. So one and other. So don't hesitate yourself to connect with the research scientist or any XYZ person is working with some reputed university or other organizations having the facilities. Connect the people and build the networking. Right. That is one thing. And I said for interdisciplinary, networking and collaboration is important. And for recognizing yourself, research contribution is very, very important, right? So that's why here I was mentioned two of the quotations to the faculty members. If you want to walk fast, you have to walk alone. If you want to walk far, that is our journey is 35 years. 35 years we would like to work as an employee. So far you have to walk. For that, we have to walk together. It means networking and collaboration. Right? Understand? So your success will be measured depending upon your hard work, your dedication, and your dreams. Okay, so that's why, my dear colleagues, like faculty delegates, start working and spend some time for your future endeavors. Thank you, one and all, for giving me this great and wonderful opportunity for sharing my expertise and experience with all of you. Okay, so if you have any type of queries, you can please don't hesitate to connect with me.
Thank
Yes,
you so
ma'am.
much, sir,
Yes, ma'am.
for such an inspiring and deeply insightful session. Uh,
Thank you, ma'am.
your extensive experience and visionary thoughts have truly enriched our understanding in innovation and excellence of herbal drugs. And now I would request everyone, if you have any query, you can ask or keep it in a chat box so that I can study that and ask from sir.
I
Any
think
question?
everyone has understood, ma'am.
Yes,
Okay, okay.
sir.
But anyhow, it's a wonderful. This is actually my 57th okay presentation, scientific session, delivering to the uh, public, like our faculty members and students in various conferences like that. So it is one of the great platform to me to share my views to national level or international level. That Gaurav sir,
Yes, sir. Yes, sir. I'm here.
yes sir, yeah, are you ready, sir?
Yes, sir. Yes, sir.
Okay,
Yes.
okay, okay.
Should I start, sir?
Yes, ma'am, ma'am, Suchita, ma'am.
I'm sir.
Sir, sir,
How?
sir, I would like to request Niranjan sir to give you the vote of thanks.
Acha, acha, okay, 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 okay,
Sir, please stop sharing your screen so that I can share.
okay. Okay, I will stop once.
Yes.
Yeah. Hmm.
Sir, I will now the I will now like to invite respected Dr. Niranjan Kaushik sir, who is HOD of Pharmacy Department in Galgotias University, to give you a token of gratitude for this contribution in today's FDP. Niranjan sir.
Yes.
Yes, sir. Niranjan sir, are you able to unmute yourself? Yeah. निरंजन देखिए निरंजन
Okay, ma'am, just you can take a note, ma'am, no issue. Okay, so because of delay, no, we have one more session.
Yes.
Yes, sir.
Am I
Yes, sir.
audible, ma'am?
Yes, yes,
Yes,
sorry,
sir.
right.
Yes, sir.
Yes, yes, sorry,
Yeah,
right.
okay,
Okay,
okay. Thank you, ma'am.
Yes,
Yes.
sir.
Thank you, Professor S.K. Abdul Rahman, sir, Professor and Dean, Department of Pharmacy, SMS, Galgotias University, for delivering such an insightful and engaging session during our FDP. Your expertise, clarity, and real-world example greatly enriched our understanding of the topic development of nano herbal formulation and its evaluation, and left a lasting impact on all participants. We truly appreciate the time and efforts you have invested in preparing and presenting the session. Your contribution has added a tremendous value of the program. Once again, thank you, sir.
Thank you very much, Nandan sir. And I thank all the participants for your nice comments. I could see in the chat box. Thank you once again. And I also thank Suchita Misra ma'am and Mamta Vishnu ma'am for the coordination of today's event. Now we will move on to second session. Ma'am,
Sir.
Yes.
go
Thank you,
ahead.
sir.
Now,
Thank you, ma'am.
thank you so much, Niranjan sir. Thank you so
Thank you,
much, Dean
Niranjan.
sir. Now, moving forward, it is my pleasure to introduce our second esteemed speaker of for the day, Dr. Gaurav Kumar, Associate Professor and Head of Department of Biotechnology, Department of Biotechnology and Bioengineering, School of Biosciences and Technology, Galgotias University. Dr. Gaurav Kumar is also the head of the institution Innovation Council and has been instrumental in mentoring more than 50 student startups. Under various innovation programs under his leadership, the IIC at Galgotia University achieved 4-star rating for 4 consecutive years. He holds a PhD in biomedical engineering from the Indian Institute of Technology, BHU, Banaras, and Dr. Kumar has published over 40 research papers, filed 14 national and international patents, authored 3 books and 8 book chapters. He has executed several funded projects as PI and co-PI. He has organized more than 200 national, international events and delivered over 30 invented talks in innovation, entrepreneurship, intellectual property rights. His research interests include neuroprotection, wound healing, drug discovery, which beautifully aligns with the theme of this FDP. Sir, it's an immense pleasure to welcome you to deliver your insightful thoughts. The platform is all yours.
Thank you. Thank you so much, ma'am, for the invitation. Uh, uh, good afternoon, everyone. And I am also thankful to Dean sir, Rahman sir, for providing me this opportunity and platform to to do the interaction with the faculty member. And can I start, ma'am? And please inform me about my timing. How much time can take?
Sir, for you, it's also a 1 hour session,
Okay.
and you can wind up with that.
Okay, okay, ma'am. So, okay, I will try to wind up at 1 p.m. Okay, can
Yes,
I
sir.
share?
Yes, sir. Sir, you are not visible in our screens.
Okay. Now, sorry.
Sir, yes, sir. Sharing is visible, sir.
Okay.
Screen sharing is visible.
I'm not able to do. Okay. Now, the PPT is visible or not, ma'am?
Yes, sir. Yes, sir. PPT is visible.
The
Yes,
PPT
sir.
is moving or not?
Yes, sir. It's full screen now.
Okay,
Yes,
it
sir.
is moving.
Yes, sir. Yes, sir. It's
Okay,
moving.
thank you. Thank you. Thank you for the confirmation. Once again, good afternoon, everyone. Today I will speak on the tools, software, database for the structure-based drug discovery with a special focus on phytochemical resources, because more than 50% of the drug approved by the agency FDA originated from the natural product. So in this 45 minutes, I will walk you through the key components of the structure-based drug discovery and how this tool enable you to do the molecular docking, molecular dynamics study, and for the drug discovery and drug discovery steps. So in this talk, we will discuss about the structure-based drug discovery, database for the drug discovery, the phytochemical and natural product databases, tools and software. The key tool I will discuss. I will not explore all the tools. AI and ML for the uh, structure-based drug discovery, case studies, one case study I will discuss, and some challenge and future perspective I will discuss. So we will begin with the structure-based drug discovery. You can see that here you can see the we have a protein structure and the ligand structure. On the basis of protein and ligand structure, we are basically selecting whether we have to do and what we have to do. So if the protein three-dimensional structure is known and ligand three-dimensional structure is known, we are generally performing the structure-based drug discovery. We are using virtual screening. We are doing the docking and scoring function. If the protein structure is unknown and ligand structure is known, so we basically the researcher want to do the ligand based drug discovery. So ligand based drug discovery include the pharmacophores, 2D and 3D, as well as virtual screening and QSR study we are doing. If the protein structure is known but the ligand structure is not known, then researcher can take the de novo designing or can do the virtual screening. Similarly, if both structure is unknown, then we have to perform a comprehensive study. We can do the study with the combinatorial chemistry. We can do the high throughput screening and virtual screening we can do. So the drug design process has two parts on the basis of the three-dimensional structure of availability of the three-dimensional structure of protein as well as three-dimensional structure of the ligand. So we can classify it into two parts, like a structure-based drug discovery as well as ligand-based drug discovery. In the structure-based drug discovery, we can choose the de novo designing, as well as we can perform the virtual screening by using several software like Schrodinger, and we can use the Autodoc and Pyrex software. In the drug design step for the ligand based drug discovery, we can use the QSR, scaffold hopping, pharmacophore modeling, and pseudoreceptor modeling. So there are several type of tools and techniques has been available to do the research. Why structure-based drug discovery matter? Because it can improve the hit identification efficiency. It can also reduce your time for the research. It will also reduce your cost for the drug discovery. You can minimize the cost for the reagent, the chemical, the protein, and it also support the rational modification of the lead and enhance the probability of the developing potent and selective drugs. So actually the drug discovery, in the drug discovery area, particularly in structure-based drug discovery, why structure is matter? Why structure matter in the structure-based drug discovery? Because if we have the three-dimensional structure of the protein as well as ligand, then we can identify the pockets. Like some molecule are binding on the allosteric pocket site of the protein. Some molecule can be competitive inhibitor, some are incompetitive inhibitor. So we have to identify the pocket, the active site, the catalytic site. We can identify the critical residue that is present within the allosteric site as well as in the active site of the protein. That helps us to to design a better drug that can perform in the in vitro and in vivo analysis also. It can also enable us for the rational design instead of the trial and error effort. So previously the bioinformatics was not implemented. Then the scientists usually performed screening for each and every chemical. But by using the structure-based drug discovery, you can minimize this step, and you can screen 1,020 lakh of the phytochemicals and other type of synthetic chemical. If you are knowing your targets, if you want, target can be a protein, means it can be a receptor, it can be a channel, it can be a semi-transport enzyme, it can be DNA, it can be an RNA, it can be a ribosome. So you have to identify your target from where your drug will be modulate and and where you want to do the docking, as well as you want to study the molecular dynamic simulation. So now you can, after performing the structure-based drug discovery, you can identify some hit molecule, and on the basis of that, for example, you have performed 50 docking or 50 structure-based virtual screening, 50 with the 50 molecule, and you got 2 molecules that has comparatively lower binding energy as compared to the a known inhibitor or any already prescribed drugs available to modulate that target, then you can compare your study with all existing drug molecule or any control molecule to identify the hit molecule, and that 2 molecule can be tested or validated in the in vitro or in vivo assay. It will also support the structure. If you have the structure of the protein and small molecule, you can perform the molecular dynamic simulation. You can do the docking. You can also use some AI based tool for the drug discovery. So from where we can get the structure? From which method? So there are some method scientists are typically using to determine the three-dimensional structure. So one is the gold standard method is the X-ray crystallography. When you will search the protein data bank, then you will get some crystal structure, some are the solution structure. So all the crystal structure has been determined by using the X-ray crystallography method. They are producing the high resolution atomic structure, and high resolution means it may be less than 3 angstrom, and the cryo electron microscope is also used to determine the three-dimensional coordinate and structure of the protein and their complexes. Solution structure can be determined of the protein by using NMR spectroscopy, and it is also used for the study of the protein dynamics. There are some AI-based prediction also privileged in this era, in this AI era, like AlphaFold 2, Roche TTF Fold. These are some software. It also assists you for performing the high quality model as well as experiment. if the structure is not present in the PDB, you can determine their structure also. So this is the workflow of the structure-based drug discovery. We are basically starting with the target identification and validation process. So if we know the target and we cannot know the target, we have to do the literature survey. So I am giving one example. For example, I want to discover a drug or I want to identify one phytochemical from the neem that can be inhibit the DPP4, or it can activate the PPR alpha. These two target are already used by the scientist and validated by the scientist that the activation of PPR alpha can reduce the non-alcoholic fatty liver, as well as it also modulate the type 2 diabetes. So you can see you have to identify, you have to do the literature survey and find out some target. So after finding the target, you can perform the molecular docking and virtual screening analysis by using structure-based drug discovery. So first you have to do the target identification. Then you can go for the structure acquisition and preparation. It can be do by several type of tools like Discovery Studio. You can use the UCSF Chimera software also. It's UCSF Chimera is a freely available software. So the three-dimensional structure can be downloaded from the protein data bank, and for any of the receptor channel or any of the enzyme. So after retrieving of the three-dimensional structure, you can prepare your library against this molecule. So in your library, you have to list all the phytochemical. If you want to work on the phytochemical, you have to list, for example, if you want to work on the Bithania somnifera, then ashwagandha, then you have to identify all the phytochemicals present in that plant, and you have to make a library, and you can search the PubChem database to identify, or IMPACT database or Dr. Duke phytochemical database to list out, to prepare your library. And you have to download the three-dimensional structure of the phytochemicals and synthetic molecule that is present in the zinc database as well as drug bank database. So you have to prepare a ligand library. Then you can go for the molecular docking and virtual scoring method. So after the docking, you can go for the molecular dynamics to study the stability of the protein ligand interaction. So you can perform 100 molecular dynamics for the 100 nanosecond or 200 nanosecond. You can also study their other type of analysis using prime by by the Schrodinger software. You can also use freely available software for the molecular dynamics simulation like GROMACS. It is a freely available and it work on the Linux system. So after the molecular dynamics simulation, you can identify your lead molecule, and that can be optimized by using the in vitro or in vivo study. And you can also perform the ADME toxicity prediction by using several servers are available to predict their absorption, distribution, metabolism, and excretion profile of any of the drug. And you can also predict the toxicity profile of the drug. So I will discuss several type of tool database related to the structure-based drug discovery that will help you to plan your study. So here you can see the major protein structure database. You can visit to the protein data bank. The protein data bank is the primary source of the three-dimensional structure of the protein. It has a three-dimensional structure of protein solved by determined by the three method previously I have discussed, like X-ray, crystallography, NMR, as well as cryo electron microscopy structure. And the protein data bank provide you the also annotated data for each and every protein. There each and every protein that are deposited in the PDB has a unique PDB ID and associated literature that can be present in the PubMed center, or you can purchase by the subscription method. That paper is very important, and the all protein has their resolution data, deposition data from which organism is used for the cloning, for the expression system. So all you can filter the PDB according to the need or to find out your suitable proteins structure. If the PDB do not have any of the protein of your interest, then you can go for the AlphaFold protein structure database, and you can predict by the Swiss model and Modbase, and Fire2 is also will help you to do the modeling and for the homology modeling as well as for the threading if the structure is not present in protein database. There are some ligand and chemical database also available. The PubChem is a freely available database. And it has a million of the small molecule deposited in the PubChem, and all molecule also has annotation. Means you can find the chemical vendor also. You can find all the identifier. You can find all the type of like canonical smile and three-dimensional structure of the molecule, the associated literature database related to their physiochemical property of each and every molecule. You can find for the one molecule. The PubChem are providing extensive information for the one molecule. You can so when you will visit to the PubChem, you will find all type of that data related to the one molecule. So there are zinc also one database. You can use the zinc also. The ChemSpider is also aggregated chemical database. So you can perform the structure search, spectral data and synonyms you can find with the ChemSpider. The ChEMBL is also an database for the bioactive molecule with curated activity data. So you can find the IC50 value, EC50 value of any of the molecule from the ChEMBL. Drug bank is also FDA having the FDA approved drugs and investigational drugs. So you can, if you want to do the drug repurposing, if you want to work on the drug repurposing, you can select the drugs prescribed for the one disease. You can utilize that drug for the treatment of the different kind of the disease. So it also includes the information related to the target, mechanism, interaction. So these all tools are very valuable, highly valuable for the your study. Next, we will discuss about the protein ligand interaction database. The protein ligand interaction database, like there are several databases already available, and we do not know about this database. So first you can visit the binding DB. It has the experimental binding affinity data. It also having the IC50 value, Ki value, and Kd value of the ligand and protein, and it cover more than 1 million protein ligand interaction. Again, the PDB bind is also a protein ligand interaction database, and it has protein ligand complexes from the PDB and include high quality binding affinity benchmarks that can be utilized to study your control molecule or drug molecule for the reference. So BioLip2 is also a protein ligand data interaction database. It is semi-manually curated ligand protein interaction database, and you can identify the biological relevant ligand here, and it will provide you the binding residue and interaction detail of the protein ligand interaction. Another database is binding mode. So you can use the binding mode, and it is also called mother of all database. So it has high quality validated binding affinity data set. It is good for the benchmarking and machine learning workflow. Now we will discuss about the prediction of ADME and toxicity dataset. So if you are predicting the pharmacokinetic data and toxicity assessment, you can perform by using in silico, by using your computer. You can predict the ADME and toxicity data of your small molecule. So there are some software that are freely available like Swiss ADME. You can visit to the Swiss ADME to predict the synthetic accessibility also. Means I endorse Swiss ADME because very few data server and database are providing the information related to the phytochemical synthetic accessibility. If you can know whether any phytochemical will be synthesized in the laboratory or not. So you can use the Swiss ADME to know the synthetic accessibility of your molecule. If, for example, you have performed molecular dynamics and molecular docking, and you have identified some hit molecule, and for example, I'm giving one example like Bithania somnifera means Sugandha has bithanolide A. And we have performed several studies using bithanolide A, and bithanolide A costs approximately bithanolide A cost approximately 80,000 for 10 mg. So the phytochemicals are available in the some chemical vendor like Merck and Sigma Aldrich, but very few quantity, very minimum quantity can take consume your huge amount of money. So you can predict if you are working on the natural compound or herbal compound, then you can check their synthetic accessibility by using Swiss ADME. Swiss ADME also provide you the ADME database, ADME property of the molecule. It can also predict the drug likeness. There are several type of log P value, log D value. You can also predict the solubility of the molecule. So Lipinski filter is there, whether the molecule can be developed as an oral drug or not. You can predict the HI, human intestinal absorption, as well as whether the molecule has the ability to penetrate the blood-brain barrier or not. You can predict using the Swiss ADME as well as PKCSM pharmacokinetic. So PKCSM pharmacokinetic is also a server that when you will provide the some structure or any canonical smiley of the small molecule, it give you a comprehensive data set for the ADME and toxicity analysis. Next is the ADME lab 3.0 or pre-admit software. I have not listed here. I have also performed several work on the pre-admit software, not software, the server. So pre-admit is also there. Admit lab is also there. Tox cast is also there. So you can use this software to predict the ADME and toxicity profile of your molecule. I am also discussing some phytochemical and natural product database like IMPACT. So Indian Medicinal Plant Phytochemistry and Therapeutics, the IMPACT database has a comprehensive list of the herbal plants, and you can it has also some annotated data like which molecule has it also provide you the citation report also. So you can check their concentration of the molecule present in the plant. You can also check whether the phytochemical is present in leaf, root, or any part of the plant. So it is a curated database for the Indian medicinal plant, and it also has the three-dimensional structure docking ready. If you will use AutoDock tool for the docking, then you can download the PDBQT file, three-dimensional structure PDBQT file for each and every phytochemicals also. That is very good type of advantage for using IMPACT if you are working on the phytochemical side. So it also excellent for the natural product based virtual screening. So you can use the IMPACT database. There are two type of search, basic search and advanced search in the IMPACT. So you can perform as per your need. Okay, so next database we are frequently using Dr. Duke Phytochemical and Ethnobotanical database. Doctor, it is developed by the USA Department of Agriculture and maintained by the US Department of Agriculture. And Dr. Duke Phytochemical and Ethnobotanical Database have the classical database for the plant-derived phytochemical and activity. There are a filter to filter out the you can exclude the ubiquitous phytochemical present in each and every plant, and you can only download the data in the Excel or CSV file for each and every plant from the Dr. Duke phytochemical database for the unique entity or unique phytochemical present in this. There are other database like NPAS, natural product activity and spice resource. So this NPAS also have the experimental activity data. The coconut, the collection of open natural product is also a valuable database. It has more than 4 lakh of the molecule, and you can predict the natural product likeness scoring here. And another database is Supernatural 2 that has more than 3 lakh natural compound database, and each and every compound can also link with the toxicity and predicted target information. Some other databases you can also use like NPACT as well as AfroDB. So AfroDB is a part of the zinc database, and NPACT is having the anti-cancer natural compound database. We all are knowing about the 8 o'clock plant that have a validated anti-cancer activity, and you can check which type all the plant resources and phytochemical that has been promising anti-cancer activity here. For the structural visualization and preparation, we are basically using UCSF Chimera. Now the Chimera has also evolved as a advanced version, so it has the Chimera X. So you can freely download the UCSF Chimera, and you can also, if you want to explore more than the visualization, then you should download the Chimera X. It will be used for the protein preparation. You can do the docking also. You can do the superimposition of the two protein ligand complex to know to quantify the RMSD, the difference between the actual PDB data, protein ligand complex crystal structure, and your docked structure. So you can do several type of work. You can do the protein preparation for the molecular docking, and protein optimization you can do. So it is very good for the interaction and trajectory analysis. You can also use PyMol. The PyMol is developed by the Schrodinger, and it has a high quality 3D visualization. And by using PyMol, you can, Dr. Prashant, why you are giving sign good marks at my PPT? Dr. Prashant, this is not expected from the faculty. This work should be done by one UG student. Dr. Prashant, if you have any query, you can ask. You can unmute yourself. Am I audible to you? If you are not interested to listen, you can move from here. Okay. Sorry for the disturbance. Next structure visualization and preparation tool is the BioVia Discovery Studio. You all must you have know about this. So BioVia Discovery Studio is also for the academic version you can utilize it free of cost. So this it also provide you advanced interaction analysis, hydrogen bond and hydrophobic mapping you can do, and this is also useful for the docking pose refinement. You can prepare your protein. You can do the energy minimization by using UCSF Chimera. Also, you can also do the 2D interaction plot you can also perform by using Discovery Studio Visualizer. Next one is the mole web-based viewer you can utilize, but there are several tools, but we have included in as a web-based viewer. Mole is a fast interactive structure viewer, and there is no installation needed in your laptop and computer. So it is ideal for the quick inspection of the PDB and AlphaFold structure. So these are the some structure visualization and preparation tool. You can also do freely use the League Plot Plus tool that I have not included here. League Plot Plus. By using the League Plot Plus, you can generate the image and you can study the ligand and protein binding interaction, like how many hydrogen bond has formed and how many hydrophobic interaction has been established by your small molecule. Next, I will discuss about the protein modeling tool. So the protein modeling tool, like Modeler, you can use the Modeler for the homology modeling and comparative modeling. It can generate the 3D protein structure using the known template, and it is useful when no experimental structure is available in the no three-dimensional structure is available in the protein data bank. You can also use, previously I have also discussed about the Swiss model. So Swiss user can use this Swiss model server to automate the homology modeling, and you can predict the three-dimensional structure. So AlphaFold currently using the AI tool for the to determine the protein structure. So this is AI-driven high accuracy structure prediction tool that useful for the protein lacking and for the crystal structure as well as NMR solution structure. iTracer is also used to do the hybrid modeling, and it also utilized to predict the three-dimensional structure of the protein. You can also predict the active site in the pocket of the protein. So it is good for the low sequence and identity case. Next are the binding site identification tool. The binding site identification tool is also needed when you do not know, for example, when you will search the protein data bank. So I will recommend you, if you want to download any of the enzyme, you must select the enzyme that has already deposited in the PDB with the co-crystallized with their substrate or co-crystallized with their inhibitor. So it will give you the exact location where you have to perform the molecular docking. So if some structure do not have any of the co-crystallized ligand with the protein, and we then it will be very difficult to you to know the binding site where we have to perform the docking, because in the whole protein you cannot perform the molecular docking. It will also not it will also not relevant, as well as it called as blind docking, and you so therefore you have to know the binding site, active site, and catalytic residue site of the protein or allosteric site of the protein to know the binding site of the protein to for the molecular docking study. So the CAST-P is a computed atlas of surface topography of the protein. You can use the CAST-P to it will help you to identify the pocket, the cavity, the loop structure, as well as surface depressions in the protein. You can also use site map. If you are using Schrodinger, then it will be very useful tool, but Schrodinger is a paid software and everyone cannot access the site map. So when you will use the site map, it will predict you for the druggable pocket in the protein. It will also analyze the hydrophobic and hydrophilic region in the protein. So it will also generate the site score and D score for the druggability assessment of any protein. You can use that F-Pocket. F-Pocket is a fast open source pocket detection tool. When you will use the F-Pocket, it will also rank the pocket on the basis of their physiochemical feature. It is also commonly used in the large scale screening pipeline. Next is the Do-G site scorer. So Do-G site scorer is automated binding pocket prediction tools, and by using you can predict the pocket of the protein. Next is the ligand preparation tool. So a protein need optimization, minimization, and refinement before the molecular docking or structure-based drug discovery. Similarly, ligand also have to prepare before the molecular docking and structure-based drug discovery. So there are several tools are available. Some are freely available. Some are on the paid basis. So the open Babel tools are freely available, and you can convert 2D structure to the 3D structure. You can convert any of the file format to any of the file format by using the open Babel software. So you can convert between 100 plus chemical format like SDF, MOL2, PDB file format. So it generates the three-dimensional structure and protonation state, and it is very essential for preparing the large ligand library. So I will recommend you to download the Open Babel because the Open Babel is freely available software. The Marvin Sketch from the ChemX zone is also it also available freely for the academic purpose. So by using the Marvin sketch, you can also predict several type of physiochemical property of the molecule. You can draw the molecule also. You can draw the 2D structure, 3D structure. You can convert one file format to another file format by using the Marvin sketch. So you can also predict the log P value, PK value of any molecule. It can also generate the NMR spectra for your small molecule. The RD kit also a tool, open software you can use for the open source and cheminformatics it is open source, freely available, and it can generate the conformer, descriptor, fingerprints, and you can use it for the filtering, clustering, and QSR feature. I have also listed here LigPrep. LigPrep has been developed by the Schrodinger and it is not freely available too. So, but by using the LigPrep, you can generate the high quality 3D ligand preparation data, and you can also predict the derive their tautomer, stereoisomers, and protonation state. And after without leak preparation, you cannot use the Desmond and molecular dynamics as well as the Glide. So Glide are the software developed by the Schrodinger. You can use the Glide also if you have the SS. Next are the molecular docking tool, and the molecular docking can be performed by a various tool. A range of the software has been available freely. Of course, there are several server also available. They will when you will upload the PDB ID and your PubChem ID of your chemical, they will perform the molecular docking and they will send you the report on your mail. But some standalone software is also there, like Glide you can use. If you are using Schrodinger, then you can use the Glide, or if you do not want to expend money, then you can use the Autodoc or Autodoc Vina. It is very freely available and developed by the the Script Research Institute. And the Autodoc and Autodoc Vina also providing you the good quality of scoring function. So it is freely used docking software. You can perform the rigid docking as well as flexible docking by using the Autodoc tool. But it will lag when you will do the protein-protein docking. So it is very compatible docking tool for the small molecule with the protein, as well as you can perform the molecular docking of the RNA and DNA structure by using the Autodoc. So Gold is also a docking software. It also uses several type of algorithm like genetic algorithm, Lamarckian algorithm, and the doc6 as well as molecular operating environment MOE doc system can be also used for the molecular docking. The scoring and rescoring, there are several scoring and rescoring method utilized by several software like scoring function. It can be empirical, it can be a knowledge based or force field based. Like if you want to study the molecular mechanics after performing the molecular dynamics, then you can study the GBSA and prime developed by the Schrodinger. You can use the prime software to study the GBSA for the generalized bone surface area. So it is widely used for the scoring method. X score is another tool, as well as MMPBSA is also utilized to study the scoring and rescoring method. The next one, the molecular dynamics simulation. The GROMACS is freely available, fast and open access software for the molecular dynamics simulation. So it support multiple type of force field like Ember, Charm, you can OPLS, you can do the molecular dynamics simulation, but it need also a high performance computer. So you must have at least 12 GB RAM, at least 8 GB of NVIDIA or any graphics card to use the GROMACS. That similarly, if you are using the molecular dynamics, then you need some workstation. You cannot perform on your simple 8 GB RAM laptop. So you must try also, because it will crash your hard drive and the loss can concur. The Ember Doc software is also there. Charm GUI interface is also developed. Desmond has been also developed by the Schrodinger, but it is for the academic purpose. Desmond of the Schrodinger is freely available. So you can install the Desmond and you can compute the molecular dynamics simulation you can by using the Desmond. So NMDA is also software you can use for the molecular dynamics study. Some AI tool I have also listed, like AlphaFold protein database. The AlphaFold are currently attracting the attention of the researcher because it due to its high accuracy protein structure prediction. So you can use the AlphaFold also, DiffDoc also, the EQBind, as well as deep generative model like de novo for the de novo design, like Reinvent, ChemTS. These all are the tool, AI based tool that assist you for the structure based drug discovery. So I have listed here one case study. I will not take more than 5 minutes for this. For example, if you want to perform, you want to identify any of the phytochemical that has some inhibitory or activation activity on any of the target molecule, then you have to select the target. And for example, if you want to work on the MMP9 to inhibit the some blood-brain barrier disruption, then you have to retrieve the structure from the protein data bank, or that structure after retrieving the three-dimensional structure of protein data bank, you must read the associated literature to know the critical residue present in the MMP. MMP is a matrix metalloproteinase enzyme, and there are some heavy metal also associated that playing a important role in the inhibition. So during the refinement, you must not delete that atom from the catalytic site. So first you have to identify the catalytic site of the protein. Then you have to build the phytochemical library, but you can use the Dr. Duke phytochemical database. You can use the impaired database to list out, to make a library of the phytochemicals present in any of the plant. then you can go for the Autodoc or Autodoc Vina. You can use the Gold also and Glide also for the molecular docking. So after doing the molecular docking, you can see their interaction. So 2D and plot interaction you can generate by the Lig plot plus, as well as you can use also the some freely available software is also there to that will predict the ligand protein interaction pattern. So the interaction analysis you can perform by using Autodoc. Also you can also use the UCSF Chimera to do the interaction analysis. Then you can also you can predict the ADME and toxicity of your phytochemicals by using PKCSM as well as the Swiss ADME tool. So we have to identify the promising plant-derived lead by using some molecular structure-based drug discovery method. So some phytochemical will show the comparable or better docking score as compared to the already prescribed drug as well as validated inhibitor or activator. So you can do the you can go for the IPR for the patent, or you can publish your research work. So there are some challenges in structure-based drug discovery also, like some molecular docking tool treat the protein as a rigid. So some researchers are also doing the rigid docking. So it will be, I am recommending that you should use the flexible docking. And like limitation of the scoring function, there are several limitations, like some software are failed to rank the compound accurately, and it has the poor handling of the water molecule, entropy and long range effect. The quality of the structure data, experimental structure may have missing loop, low resolution. So predicted structure like from the AlphaFold may require the refinement. And if you are predicting from the modeler and any other of the tool, then you have to perform the molecular dynamics simulation. So like incomplete chemical space coverage, for example, if you want to list out or make a library on the synthetic library, so it is very limited, and some database of the small molecule are paid. So there are some challenges in the structure-based drug discovery also associated with this. For example, I have some, I am showing you some research paper that has been published in the SCI journal. And you can see we have published some research by using only molecular docking study. We have published these all research that is listed in the SGR list. And now I am thankful to the organizer for providing me this opportunity. Thank you everyone for listening. Thank you, participant. Thank you so
Thank
much.
you so much, sir.
Yes.
Sir, now for a quick Q&A session.
Yes.
If
Yes, ma'am.
anybody wants to ask any question, either you can post on the chat.
Yes, or you can unmute yourself to discuss.
Yes.
Oh, some question is coming in the chat box
Yes,
also.
sir. Yes, sir.
One question is, sir, in my protein, Arpan is asking, sir, in
Arpan
my
Pathak is
protein,
asking.
yes, contain 3 chain, that is A, B, C, and priority discovered ligand binding site in chain A and C. But I want to bind my ligand in chain B. Why you want to do the docking in chain B? Actually, I have already discussed that whenever you have identified your target molecule, you have to learn you have to go through thoroughly with the associated literature data with that protein. Some protein, for example, NMDA, are having several chains, and some cross chain are making a dimer, or it can that both chain A and chain C can be utilized for the molecular docking. So you have to know you have to know the structure first. You must know all the aspect of the binding pocket of your protein. So you cannot remove any of the chain. If you have downloaded, for example, if you have downloaded any of the protein like DPP4, for example, then it has a chain A and chain B, and both ligand has been bonded in chain A and chain B, means, and you have to do the comparison whether both chain are isomeric or not. If both chain are the isomeric, then you can remove one chain and you can perform the docking with the chain, either with the chain A or either with chain B. So you should not remove, because you have not discussed here which protein you are taking. So you have to know, you cannot remove the heavy atom also that is present in the catalytic site. For example, NOS, nitric oxide synthase, has a heme domain in their catalytic pocket. You cannot remove the heme domain from their catalytic site. In the MMP9, MMP2, other type of matrix metalloproteinase that has a heavy atom in their catalytic site, you should not remove that because the magnesium ion or magnesium ion should needed for their catalytic activity. So be careful during the optimization of your protein and preparing your protein structure for the molecular docking. Okay, so I think you got
Yes,
it.
ma'am. Nikhat Khan, do you have any question? Ma'am Nikhat Khan?
Any other?
Is
Okay,
there any
PPT.
other
I will share
question?
the PPT with the organizer, and they will send to you.
Yes, sir.
Okay, thank you, thank you.
Sir, now I would like to invite Madam Dr. Mamta Vishnoi to present you a token
Welcome.
of appreciation for us. It's a very appreciative for you to take your time from your busy schedule and give such a informative session for us. Ma'am Mamta
Thank
Vishnoi,
you
ma'am.
so much.
Thank you, Suchita ma'am. Thank you so much, sir. Thank you
Thank
so
you,
much
ma'am.
for your highly informative and thought-provoking session. Your insights into modern approaches to drug design and development have truly enriched our today's FDP. On behalf of the Department of Pharmacy, School of Medical and Allied Sciences, Galgotia University, I take this opportunity to express our heartfelt gratitude and present this certificate of appreciation as a gratitude and present this token of our respect and thanks for your valuable contributions. We are truly honored to have you with us today, sir. Thank you once again for sharing your time and expertise with our participants. Thank you once again, sir.
Thank you once again, ma'am, for inviting me, and I once again expressing my gratitude toward the Dean, sir, who has providing me this opportunity. Thank you, ma'am. Thank you, the
sir.
organizer, all the organizer, and all the participants. Thank you.
Thank you so much, sir.
Thank you.
Now I'm providing the feedback link for all the participants in the chat group. You can access it and give your very valuable feedbacks to us. And these feedbacks will be counted as your attendance for the day. I also provide feedback daily in your chat box of YouTube. You can access it from there also. For the PPT, you can personally mail to the speaker, whoever is speaking here. In the brochure you have got for the registration, it must be mentioned the name and the college of the speaker. So you can contact them personally as it is their personal, uh, copyrights to have it with them. So you can ask them personally if they are willing to give it to you. They will forward it to you on your mail. I hope you understand. Whoever has got the FDP feedback can leave the meeting. I am just lingering the meeting so that everybody can be able to access the feedback form.